Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the biology course of class 11 and 12 under the IIT PAL program. And in this series, we were discussing about the chapter Biotechnology, Principle and Processes. And if you recall what we have discussed in our previous lecture, how the biotechnology is utilizing the two different principles. Either it is utilizing the genetic engineering as a tool to generate the genetically modified organism, or it is utilizing the bioengineering or bioprocess technology to enhance the production from the organism. So while we were discussing about genetic engineering, we discussed about that the genetic engineering relies on the tools as well as the techniques. And within the tool, we discussed that the biotechnology depends on to the some of these major tools like polymerases, restriction enzymes, as well as the ligases and the vector. Whereas in the case of procedures, if you want to generate the genetically modified organisms, what you are supposed to do is first you have to isolate the genome of the particular organisms, and then from the genome you have to use. So first procedure what you have to learn is the isolation of the genomic DNA, and then with the help of the polymerase enzyme you can be able to isolate the gene, and that you have to achieve simply by going with the polymerase chain reactions. And once you got the gene, you have to cut this gene with the help of the restriction enzymes. So the restriction enzyme, you have to perform a restriction digestion reactions, and then once you done the restriction digestions, it is actually going to generate the sticky end on the both side of your gene. With the same way, you have to also isolate the plasmids. The plasmids are the vectors which are actually going to carry the gene of your interest. Then you have to restriction digest the plasmids so that it is also going to generate the sticky ends. And now you have to take the cut gene as well as the cut plasmid and put them together within the ligation reactions. So you have to learn also about the ligation reaction. Now, once you have generated the sticky ends like here, right? Then you have to, and if you put them together, they are actually going to form the complex, which means because of the complementary sequences. But on the edges, they are actually going to have the gaps, and these gaps are going to be sealed by another enzyme which is called as the ligase. So the ligase as the name suggests, it is the enzyme which is actually being used to join the DNA. So if you have the two DNA and you put them together with in the presence of ligase, it is actually going to ligate the. So the ligase is like a glue, right? The glue what you use in your home for sticking the papers, right? So ligase is like a glue which actually going to glue the two DNA sequences. So ligase joining the two DNA to generate the chimeric DNA is the basis of the cloning. It is an essential step to generate the cloning containing the foreign DNA in a vector. When the cohesive end generated by the action of the recession endonuclease on the DNA associated with each other, a nick remain to seal and give the complementary DNA sequence. Which means what happen is that if you have this particular type of vector and it is actually generated nick on one side and nick on the other side. So what will happen is you are actually going to put the foreign DNA, but on this side, as well as on this side, there will be a small gap, and that gap will be a gap between the OH of the one DNA versus the phosphate onto the other side. So there will be a bond missing between the OH and the phosphate, and this bond formation is the reaction which is going to be catalyzed by the ligase. So what DNA ligase is doing? It is an enzyme require ATP or NAD plus as a cofactor to catalyze the ligation reactions. Ligase is processing ATP to generate the AMP or the adenine monophosphate, and then the AMP is making an adduct with the enzyme to form the ligase AMP complex. This complex is binding to the 3' and 5' of the DNA, bearing nick and bringing them together. AMP is released and the phosphodiester linkage is formed between 3' and 5' end to seal the nicks, which means it is actually going to form this particular type of bond. So you can imagine the ligase is processing the ATP and generating the pyrophosphate or PPI, and in that case the AMP is sticking to the enzyme, and then this is the bond, right? You have a OH on this side and you have a phosphate on the other side. This bond is missing. So what happens is the AMP will go and bind to the both sides, and then it will bring them together, and ultimately the AMP will be released and this particular type of bond is going to be formed. And that's how the two DNA is going to be linked to each other. Now, I think we discussed about the setting of the recession enzymes. Similarly, you have to set up the ligation reactions. And remember, here what we are doing, here we are doing exactly the reverse what we were doing for the recession enzyme. So in the case of this, what we are doing, we are putting the two DNA, like DNA 1 and 2, and generating one DNA, right? Which means this is actually going to give away a water molecule, which means, and this is being done in the case of ligase, right? So there is a dehydration reaction, which means this one and this one is actually going to lose the hydroxyl group as well as the H plus, and that's how it is actually going to generate the DNA. So apart from that, this DNA and this DNA has to interact very extensively, and because of that, the ligation reaction has to be set up in a very small volume so that it is actually be allow the molecular crowding, and because of that the ligation is going to be more and more efficient. So technically we always set up the ligation reactions in a volume of 20 microliters so that the enzymes as well as the two DNA what you are adding, whether the vector DNA or the insert DNA, should interact extensively. Now, we have discussed about the polymerases, we discussed about the recession enzymes, we discussed about the ligases, and all these are working on the two DNA sources. One, we are working on the gene, which is we are isolating from the genome and generating the sticky ends, whereas the other is the plasmids. So before we discuss about further, this DNA has to be integrated into a plasmid or the vector, because the vector is the only DNA which has the ability to carry this DNA into the host cell, and that's why the vector is very important for the complete process of the genetic engineering. And vector is a very important tool because it allows the carrying of this particular DNA into the host of our choice. Now, vector, what is the two function the vector DNA has to perform? It has the ability, it should have an ability to carry the foreign DNA, which means the DNA what you are interested to put it into the cloning reactions, and the second is the ability to replicate in the host. Hence, to fulfill these responsibilities, a number of properties are desirable from the vectors. But before discussing about these properties, let's discuss about the different types of vector what you can use. You can use the cloning vector which is being used for the cloning of the DNA. So what is the use of the cloning vector? The cloning vector is being always used the places where you are either interested to study the particular type of process. For example, if you are interested only to study the DNA replications, uh, so in these cases you can use the cloning vector, or in some cases you are not interested to express or do something, but you want to have the you want to generate the DNA, and you want to insert that into a vector so that it should be you can be able to have a complete set of DNA, and that you do simply to generate a library. So you want to generate a library of DNA into a vector, and that is called as the genomic library, for example. So if you generate the genomic library you can generate into a cloning vector. And what is the purpose of this genomic library? This genomic library you can use if you want to do, for example, the DNA sequencing. So, for example, the people have prepared the genomic library of all the genes what or the all the DNA what is present in the human genome, and that's how they could be able to generate. They could be able to sequence the complete genome. For example, in this case, what you'll do is you actually are going to first generate the genomic library. So, for example, if I have a library in my home, for example, a book library, I can actually do a cataloging. For example, I can write this is the clone number 1, this is the clone number 2, this is the clone number 3, and this is the clone number 4, and so on. So that's how you can actually represent the complete genome into a genomic library by having the gene into the cloning vectors, different part of the genome into the cloning vector. And then what I can do is I can just sequence this 1, 2, 3, and 4, and then I can put them together, and that's how I can have the complete genomic sequence of that particular organism. So because if you try to do that the same way, if by isolating the complete genome, it will be very difficult because the size of a particular genome is going to be very big, whereas the size of the DNA in this particular small clones are going to be still be manageable and can be sequenced accurately. So this is one of the application of the cloning vector. The other is that you want to study some basic processes or some activity of some protein and all that. So that way also you can actually be able to use the cloning vector for studying, for example, DNA replications and transcriptions and all that. Uh, Apart from that, you can also use the expression vector. So the purpose of expression vector is to produce the protein from a particular gene. So in this particular case, you can actually be able to clone a particular gene, and that's how you can be able to use that for cloning purpose. Now, what will be the criteria of a good vector? So before we discuss about the components, what is present in a good vector, you can also think about the criteria. A vector should be of small molecular weight, which means the low molecular weight of size confers a number of advantages. For example, the small vector is robust toward the shear stress and easy to handle. In addition, after the ligation, the foreign DNA into the vector, the size of the resulting recombinant DNA will be small, and it will be easier to deliver the recombinant DNA into the vector, which means you can imagine this very simply, that if you want to handle or if you want to send a packet right from your hostel room to your home, and the packet, if you want to send a packet, you don't want the packet to itself be of 100 kg, right? You want a packet to be as light as possible, so that because once you reach to the postal office and you want to send it, and suppose your letter is only 1 kg, right? Then the total will be 101 kg, right? Whereas 100 kg is actually a waste, because 100 kg is your packet. But if I imagine that if I only have a packet of 100 grams, then the total is going to be 1.1 kg, which is actually of 10% of this. And sending this is going to cost less compared to the sending of this particular type of packet. So that's why it is always desirable that you should have a vector of a very small molecular weight. The second is, which is very important, is that once the vector enter into the host, for example, the bacteria or yeast or mammalian cells, it should give us some kind of phenotypic changes. Another important feature is that the vector DNA should give the additional phenotypic changes in the host cell so that the recognition of the transformed cell will be easier. Which means if this is the cell and this is the cell which actually got the vector, it should have some kind of phenotypic changes, so that just by looking at the cell or just by looking at the appearance of a colony or by employing some kind of screening criteria. For example, if this particular cell, by having this particular type of vector, acquire the resistance against the ampicillin, right? So if I put these two cells, which is this is the untransformed cell, this is the transformed cell, under the ampicillin, only this cell is going to grow and this cell is going to die. So that is a very easy way of identifying who got the recombinant DNA versus who didn't, right? And that is very, and for example, there are other criteria is that, for example, if it is actually going to give some color, right? So if it is started showing some color, I can easily see that, okay, the red color cells are actually the recombinant DNA containing cells. The blue color cells are non-recombinant DNA containing cells, so they are useless, right? So that actually is make the process very easy. The third is you should have the multiple cloning sites. So what is mean by the multiple cloning site is the multiple cloning site is a stretch of DNA which actually contains the repetition sites on a regular interval. So they have actually a very composite DNA, and all this DNA is actually going to have the repetition enzymes. So you can have the flexibility. For example, this is 1, 2, 3, and so on. So you can have a flexibility of using the first excision enzymes and the the middle one, or you can have the third excision enzyme and the last excision enzymes and so on. So a small stretch of DNA on a vector DNA containing the excision site for possible site of insertion of foreign DNA is desirable. So if you have multiple numbers, you have more and more flexibility of choosing 1 and 3 or 1 and 4 or 1 and 8 or something like that, because all these have to be chosen as per the set of criteria what you have, what kind of registration enzymes are not present on your insert DNA. Now, for high copy number, it should have a high copy number, which means if I grow this particular type of vector, I should, and same amount of cells, I should get the more amount of DNA, which means the yield of DNA is going to be more if I have a high copy number vector. And since the yield of DNA is going to be more, it is actually also going to give me the more amount of protein if it is a high copy number vector with a expression vector. Now this is all about the criteria of a good vector. Now let's discuss about the different types of component what you require. So components of a particular typical vector. There are minimum molecular component you need to assemble for a bacterial plasmid to perform the function of vector. For example, so I have taken an example of bacterial plasmids. You can imagine that the components will remain the same, except that you might have to take those components from the respective DNA. For example, what the first component and the most crucial component is the origin of replications. What is mean by origin of replication is that in a bacterial plasmid or the bacterial chromosome, it actually starts the replication from one end, and then this continues like this, and it actually goes to complete. This place from where the replication starts is called as the origin of replication or the ORIC. Like any other replicating DNA, the plasmid DNA needs its own independent origin of replication to provide the replication start site to make the copies. It decides the range of bacterial host strain can be used for a particular plasmid vector. So depending on the type of the origin of replications, you can be able to choose the host. For example, if it is a bacterial origin of replication, you cannot use the yeast or mammalian cells to replicate this particular DNA, because the host is actually going to provide the machinery to recognize this origin of replications. So if I have a bacterial plasmids, like for example, it will not going to replicate into the mammalian system, because the mammalian system is going to provide the DNA polymerase or helicases or SSBs, which are actually not going to recognize this particular DNA. You require the DNA polymerase and helicases and SSBs and tethering proteins from the bacterial origin, so that it should be able to recognize by those particular type of machinery, and it should replicate. And within the bacteria also, it may be restricting to gram positive versus gram negative. For example, the plasmid containing the origin of replication from col E1, this is a kind of a can be able to grow in limited bacterial species such as E. coli. In contrast, the plasmid containing origin of replication from RP4 or RSF101 can be able to grow in gram negative as well as the gram positive bacteria. So depending on the type of origin of replication you are going to have into the plasmids, it is actually going to restrict the replication of that particular vector or that particular plasmid into the bacterial species. For example, if you have col E1, it is only going to replicate in E. coli, whereas if you have the original replication from RP4 or RSF101, it will be more relaxed, so it will actually going to grow in most of the bacteria, whether it is gram positive or the gram negative. Then you require the selection markers. The selection marker in the form of either antibiotic resistance genes or enzymatic gene is essential to keep the phenotypic changes in host after the entry of the plasmid. So this I think we discussed. If you have the selection markers, you can be able to use and calculate, you can be able to use that as a criteria to choose which one is non-transfected versus the transfected DNA. So for example, if I put the ampicillin, this one is not going to grow and this one is actually going to grow. So that's why the colony what you are going to get is actually going to have this particular type of DNA. You can have the enzyme also. In some cases, it's not that antibiotic resistance gene is present. In some cases, the enzyme is also present. In that cases, the enzyme is actually going to give you the enzymatic product like the colors and all that. So that also can be allow you to identify that transformed DNA and transformed host cells. Then the third is the promoter. So the promoter is a region which actually recognizes and which actually allows the binding of the expression machinery. Like for example, the promoter is actually going to allow the binding of the RNA polymerase and the all other kinds of if you have the promoter is always present onto the expression vector, and it is not a component essential for the cloning vector. So for cloning vector, you require only two components, like the origin of replication as well as the selection markers. Whereas if you want the expression vector, then for the expression vector, you require the origin of replication, selection marker, as well as the promoter. So the plasmid replication in yeast is performed by the host provided proteins such as DNA gyrase, helicase, polymerase, and DNA ligase. But the protein required for conferring antibiotic resistance or enzyme used for selection transform is present onto the plasmid, and promoter adjacent is required to express the gene present on the plasmid. In addition, promoter is also needed to express the gene present onto the foreign DNA. So the promoter is something what is required for the expression plasmids. Now, as I said, you can have the different types of vectors. So the vector need to be replicated in host strain, and vector need a special additional structural feature to make it suitable for a particular host strain. Why one vector does not replicate in different host strain? We have discussed that, right? The replication of a vector DNA is controlled by the origin of replication, and it need to be recognized by the host factors, especially the DNA polymerase, to perform the replication. Consequently, there are different types of vector DNA to suit the cloning of a foreign DNA in a particular host strain. So you can have the bacterial plasmid, you can have the phage-based vector, you can have the yeast vector, and you can have the mammalian vector. So you have a mammalian vector, it is actually going to replicate into the mammalian system, right? Mammalian means the all the the normal cells. The yeast is actually the yeast based vector. Then you have the phage based vector, which is actually going to work, and then you can have the bacterial plasmid, which is actually going to work in bacterial system as well. So this is a important plasmids which is actually been discovered, or the first been synthesized by the man-made plasmids. So PBr322 is a first man-made plasmid where the scientists have acquired the different region of the DNA from the naturally occurring plasmids. So these are the naturally occurring plasmids from where the scientists have isolated a different gene, a different region of the DNA, and that's how they assembled them and to synthesize the first bacteria man-made plasmid, so which is called as the PBr322. So they have taken this region from the PSC 101, which is actually a plasmid, naturally occurring plasmid in a bacterial species. Then they have taken the origin of replication from the PMB1 derived material. So the origin of replication from this particular plasmid, that is also a natural plasmid. And then they have taken the ampicillin resistance gene from the RSF2124, and that is also a naturally occurring plasmid. So what you see here is that it has the two gene antibiotic resistant genes. One is ampicillin resistant gene, the other one is tetracycline resistant genes. This region, the tetracycline resistant gene containing region, is being taken from the PSC 101, and that's how this plasmid is can be used for the cloning of the foreign DNA, and it can be select either using the ampicillin as a selection marker or the tetracycline as a selection markers. So this is all about the different types of tools what we are using in the genetic engineering. What we have discussed about the polymerases and how the polymerases are useful in terms of the isolating the particular type of gene from the genome, and how the once you got the gene, how you can be able to use the recession enzyme to generate the DNA fragment with the sticky ends. So you have the two sticky ends on both the sides, and that can be used in downstream ligation reactions, which is being catalyzed by the enzyme ligase to generate the chimeric DNA. And you can have the this particular process either the directional cloning or to the non-directional cloning. So directional cloning is always easy, and it gives the more reliable results compared to the unidirectional clonings. And apart from that, we also discuss about the different types of vectors and what are different components required to assemble a vector and can be used. So vector could be of two different types, a cloning vector or to the expression vectors. And what are different components required? You require the origin of applications, you require the selection markers for the cloning vectors, whereas you require the original replication selection marker as well as the promoter for the expressions vector. And at the end, we have also taken an example of PBr322 and how the scientists have developed the PBr322 utilizing the different DNA fragments from the three naturally occurring plasmids and from the different bacterial strains. So with this, we would like to conclude our lecture here. In the subsequent lecture, we are going to discuss about the different types of procedures. We are going to discuss about the isolation of genomic DNA. We are going to discuss about the PCR, recessionations, ligations, competent cell preparation, transformation, as well as the screening of the clone, so that you will be able to see that the recombinant DNA is being generated. So with this, I would like to conclude my lecture here. Thank you.